Okay. Today my topic is exploring complex networks, and as you can see in my outline, I have surveyed several papers. Yeah. And because I think I have a basic direction to find the paper. So, but they all have a relation about the network. So I picked up one paper as my title, and I delete three of my surveyed paper because they are not highly relevant to my topics. So you can see I picked 5 papers as my report content, and the first paper is the human connectome, and you can see that what is the connectome, and if everybody listen to a teacher, you will understand what is the connectome. That is we want to discover the human brain. So first step we need to understand the connections and of the structural structural substrate of our brain. So the purpose of the connectome project is to build up a connection matrix of the structural elements like neurons and connection paths and edges. So the first paper summarized some challenge and impact of the human connectome. So we can see that it plays out. There are two main challenges. The first one is the brain is highly complex organ, so it is difficult to define which label. We may define the labels of our brain because it is too complex. And the second challenge is the basic structural element is hard to define. Yeah, so because some function or some behavior is target to different labels, like it may provide regions to elements like neurons. So it is hard to define. But he still plays out. We may go through this project to divide the brain into three labels: the micro scale and the macro and meso scale. So the micro scale is single neuron and synapses. That is we may call it essence, or the basic elements of our brain. And the macro scale is the brain regions. Many neurons and synapses and the connections they clustered and make some functions. So we can see the brain in this label. And the mesoscale is a scale between micro and macro. So it is defined as some groups of neurons or some population of neurons. That is, the scale is smaller than a brain region. and they may have some patterns between each other and connections to other neural populations, and the authors suggest that we should focus on the cortical thalamic system first in the Phenaton project, and because I think we now notice the cortical thalamic system more than the other two types networks, and yeah, and I see the when I search for the papers of 3 types of networks proposed by the Edelman, lots of papers are focused on the thalamic cortical system, and yeah, the ratio between these 3 types networks are, yeah, I see most of the papers are about the thalamic cortex. Yeah, but I think if we the next step of the kinetome must be to the whole brain, so the other 2 types must be surveyed or studied soon. Yeah.
You may use naturally to explain the term.
Okay.
For the cortical system, I think.
Cortical thalamic system 就是视丘跟大脑皮质的连接。那我上一次的报告就是在讲这一个部分，就是thalamocortical motif。那大家可以把那个thalamus 就视丘当成一个 gateway， 就是它是把，假如说我们人身体会有感觉嘛、感知嘛 perception， 那所以这些感觉呢，3你有听懂吗？这些感觉呢，就会借由神经传导，经由 spinal cord 就是我们的脊髓，然后往上到我们的 thalamus。那 thalamus 当做一个 gateway， 它会发散 projection， 一个扇形的projection 到我们的大脑皮，就是 cortical。所以这就是一个人家Edelman 他提出一个 define 我们大脑里面3个网络其中一个网络是这一个。对，那对，那因为 cortical 就是皮质上面有很多区，每个区它有不同的功能，所以说你不同的感知上传以后，它会上传到不同功能，例如说它耳朵听到的、眼睛看到的，它会传到 cortical 里面，就是大脑皮质不同负责这个感知的区域这样。简单的介绍是这样。Okay, and then the author also suggests the 8 steps toward the human connectome. As we can see, the first step is diffusion weighted image and also wide structural probabilistic connective matrix. That means we have we use functional MRI or some other techniques to get the weighted image of our brain, and then we build up the connection matrix of the structural connection matrix. We can, as we can see here, like this one. Here is a matrix that they have the connections. Yeah. So, and the step 2 is correlation analysis of spatial register, and that means we define the several regions in our brains, like Broman's area.